Okay, so the title of my talk is Quantitative Stratigraphy from the CONOP to the High Resolution Geological Time Scale. And we are quite familiar with these figures on the left. That's a geological time scale published by International Commission on Stratigraphy. So it has been updated every year. And so that's a traditional method we used to create our geological time scale. And the resolution we can get from that method is about 1 to 10 million year. So one important product from that is Paleozoic diversity pattern published by Sapkowski in 1981. So you can see that there is a big five-mass extinction and also two major radiation in the past 600 million years. But because of the resolution of the geological time scale, so the diversity pattern on the right, the resolution is also about 8 to 10 million year because the basic the global stratigraphic correlation resolution is quite lower. And there come out one significant scientific question: why the time scale difficult to build? How can we increase the temporal resolution of the global stratigraphic correlation and also the time scale? There are several reasons for that difficulties. One is that different localities, for example, they preserve different taxa. So this figure shows 19 different sections from South China, and each section represented by a column here. So it contains many species from the base to the top, and some of them they are quite short. And each horizon line indicate the first appearance or the last appearance of one taxa. So we can see that different section, different locality, they preserve different taxa. And also different localities, they may preserve different sequence of the taxa. So you can see that some of the horizontal lines, they are crossover with each other, they are messed up. That's because some of the first appearance of the taxa, some of the taxa appear a little earlier in one section, but appear later in another section. That's because of the sampling bias in our paleontological study and stratigraphic study. So generally, observed stratigraphic section contradict with each other. For example, these two sections in detail regarding the sequence of the first appearance and the last appearance of the text of the same taxa. For example, the pink one and the blue one, so indicate two different taxa, but their sequence of their first appearance and last appearance, they are different. So that's a problem why the time scale difficult to build if we want to use a fossil to help us to construct the time scale. So the traditional biostratigraphic work, we try to find out those taxa that has a consistent occurrence through the study area. For example, this is the raw data. We have hundreds of taxa from those data sets, and we will try to find out maybe about 5 to 10 species. They shows consistent through these study sections. So if we link them together, they didn't mess up with each other. So we take them as an index fossil and use them to establish a biozonation for this study area and interval. So that's why the temporal resolution decreased from the raw data to only 1 to 10% after the traditional biostratigraphy, because we only pick up 1 to 10% of the taxa to establish a correlation. So is that possible for us to use all the raw fossil record to establish the stratigraphic correlation? Yes, it's possible. So that's why we have the quantitative biostratigraphic work. So the quantitative biostratigraphic work, they try to resolve the contradict among different sections. They try to adjust the first appearance or the last appearance of each taxa and make them consistent at each locality. And after such a adjustment, you can see that all the the first appearance and last appearance event, their order become quite consistent among different localities because they keep all the data, all the text information during the calculation, so they can keep the temporal resolution of the original data so they can keep the same temporal resolution after calculation. So that's why the temporal resolution of the quantitative stratigraphy can be 10 to 100 times better than the traditional biostratigraphy. And so there is a method which called CONOP, which has been designed by Peter Sadler in 1990s. And it provides very high resolving power for the stratigraphic correlation, but it requires much more computing power. For example, for 10,000 species dataset, that's what I used in my 2020 science paper. One calculation will take about 17 years for just one calculation. But the temporal resolution can increase from the traditional 8 to 10 million year to about 20 to 30,000 a year. So that's a big increase in the temporal resolution, but require a very large computing power. So this method, as mentioned, designed by Peter Sadler from UC Riverside in 1990s, and he published one significant paper in GSA Bulletin in 2009. So the title is High Resolution Early Paleozoic from Observation to Cellular Time Scale. And using the same method, they use a CONOP to study about 400 sections and 1,900 species from the world. That's all guacanites. That's a marine invertebrate for the observation and the cellular. And this shows the global stratigraphic correlation based on CONOP. So each vertical column indicates one section from different continent. So you can say, for example, these sections come from China, and these sections come from France. And so the vertical axis, that's time from observation to cellular. And the horizontal axis indicates the space from different continents. So you can say after the calculation of CONOP, all the section has been calibrated to the geological time scale, and the temporal resolution of the geological time scale is about 30,000 a year. And so why it works can work out such a good temporal resolution? Because through CONOP, we can convert the stratigraphic correlation problem into a sequence optimization problem in computer science. For example, we have two localities, section A and F, and we have two taxa, the red one and the green one. And you can see that their sequence, the order of their first appearance and last appearance are not the same. And from the computer science viewpoint, there are only 6 possible sequences of those two species. For example, the green one appear before the red one, and the green one before the red one, but they have overlap. Or the green one, the range larger than the red one, or the red one range larger than the green one, or the verse. And so there are only 6 possible timeline sequence of the first appearance and last appearance of the two taxa. And so we try to find out the sequence with the least adjustment. For example, among the six sequence, if we adjust the original local observation at the two sections to match with each of the sequence on the left, for example, for the sequence number 4, we just need to extend the red one, the first appearance of the red species, a little downward before the first appearance of the green one, and then the two the taxa from the local observation of the two taxa on the two sections, they become consistent. So that means the first appearance of the red one, and then the first appearance of the green one, and then the red one's last appearance, and then the green one's last appearance. So they become consistent, and also become consistent with the sequence number 4. And we call this range extension as a net adjustment if the local observation need to be consistent with sequence number 4. So we can do it with each of those sequence, and we can find out actually the sequence number 4 provides the least net adjustment for the starting data set. So that's very simple computer science resolution for this geological question. And so we started this project in 2013, and I visited Peter Sadler, and we worked together. We designed the first .NET CONOP version, and after 2 years, we make the program about 33 times faster than the original one. And finally, we found that the data set we want to analyze is much bigger than the data set that Peter Sadler has been studied before. For example, in 2009, the GSA bulletin paper, Peter only studied about less than 2,000 species, but our data set is much larger than that one. So it's at least 11,000 species. So it requires how to say the computing power about at least 1,000 times faster than the previous one. So we started the supercomputer plan in 2015, and we spent about 2 years to make it available on the supercomputer in China. And when we use 10,000 CPU cores, the program can be about 5,000 faster than the original CONOP program designed by Peter Sadler. So this is the first tool we designed and make it available in 2017. And also that's the first program for the big data analysis for the stratigraphic correlation. And when we have that program available, so we can use it for some scientific study. And where the data come from? So we use the data we digitized into our one stratigraphic data platform. And it contains now about 2 million fossil records from about 26,000 sections from the world. So here shows the geographic coverage of the data record, and all the data open to the public. So everyone, they can visit the website and download the data they want to use. So here shows the link of the website. And this is the first result we got based on the new CONOP Saga program. And you can see that it studied the data set about 11,000 species from 3,000 published stratigraphic sections. So all the section has been published. So that means they already have the passed peer review procedure for the target journal. And so here shows a biodiversity curve based on the high resolution time scale we created using CONOP program. So if we compare it with the previous study, for example, John Arroyo in 2008, they published a very important Paleozoic marine invertebrate diversity history based on the global about 200,000 fossil occurrence. And the blue one shows their data point. So there are only 25 data point for the Paleozoic. And our biodiversity curve, it contains about 11,000 data point, the red one. So you can see the resolution increased from 10 million year to only 26,000 year. So that's about 400 times better than the previous study. And we can get some more details about the evolution history of the marine invertebrate during the study interval, including the Great Elevation biodiversification, the end of ocean mass extinction, and also the end Permian mass extinction. And also we can, because we have the high resolution for our curve, so we can try to decrease our resolution curve to different temporal resolution, for example, like 0, 1 million year, 1 million year, 2 million year, 5 million year, 10 million year being, and also the PBDB time being. So you can see that all those even and divided time being from 0.1 to 10 million year has shown in this map as a solid line, and the dashed line, that's a PBDB timeline they used for the 2008 paper, because it's not really evenly divided timeline. So I use a dashed line to indicate them to indicate that curve. So you can see if we reduce the temporal resolution of our curve to different temporal resolution, then you can see some of the biodiversity change pattern disappeared or even become opposite. For example, you can see that some of the decline of the biodiversity valley here. I mean, for example, on the PBDB timeline, it become obscure or even become a little higher. So that's why the temporal resolution really matters if we want to study the long-term patterns in the Earth history. So that's the first tools we designed from 2013 to 2017. And in the past 3 years, we try to improve our program, make it more efficient and better than the previous one. So recently, we designed a new CONOP program, which called the CONOP EA. So EA comes from the that indicates the evolutionary algorithm we integrated into the program. And that's also a very important AI algorithm that can be used for the stratigraphic correlation. So the basic idea is that we just integrate the principles of evolution into our program, including the natural selection, recombination, and mutation. So, for example, the algorithm will try to evaluate each individual's fitness. So that means the solution, the sequence I listed in the previous slides, and try to select two individuals. That means two different solutions, and make them recombined together. And so that means, for example, we maintain a population with about 1,000 individuals. Each individual indicates one CONOP solution. Some of them, they are very good. Some of them, they are maybe not that good. But we put them in the population pool, and we randomly pick up two individuals, just like two DNAs, and we do some crossover of the two DNA. That's called recombination. And then we introduce some mutation, some minor change on the result sequence to that means the offspring, and we do this process many times, and we also evaluate the fitness of each offspring. So if the offspring become better than the father or the mother, we put them into the population, replace the father or the mother. So we just keep the population upgrade after each calculation. We also keep the best 1,000 individuals in the population. So that's a basic algorithm we used in this program. And because so you can see that the blue one, the figures on the left. So you can see the comparison of the two programs we designed. The blue one, that's a program we designed in 2017 and published in our 2020 science paper. And the yellow one, that's a new one. So you can see that the penalty, that's a description of the performance of the program. So if the penalty is lower, that means the program is better. So you can see it can be about 10% better than the previous program. That's because the CONOP EA program can dig more deeply in the solution space. Why? Because through the recombination, CONOP EA can combine solutions from different local solution space. For example, this is local solution space. They can combine the solution from different local solution space to generate a new solution with excellent ability to escape from the local solution space to jump into the global minimum solution space, so it can dig more deeply into the whole solution space. That's why it performs much better than the program we designed before and also the original CONOP program. And we just recently used that program in a new study on the using oligocene transition. So we studied the marine diversity curve of the foraminifera from 24 million years ago to 20 million years ago. And the temporal resolution of the stratigraphic correlation is about 29,000 years. And we can recognize for major biodiversity change in the study interval, including two diversity increase and two diversity decline. And each of them represents a significant influence of the environmental change. And based on that biodiversity curve, we can correlate with the environmental proxy data, which is also high resolution for the Cenozoic. And we can find that the different ecological group of the foraminifera, including for example, the pandemic foraminifera and the large benthic foraminifera living in the ocean surface or the warm shallow environment, they are very correlated with the ocean temperature and the sea level change. And the small benthic foraminifera living in the deep ocean, they shows clearly correlation with the carbon isotope data. So this is the study we just made based on the new CONOP program and the high resolution time scale for the Cenozoic. And so when we can use different program to establish the high temperature resolution time scale, and so another question that can we correlate other geological record to the high resolution time scale, so we can improve our previous study, to a high resolution time scale? And the answer is yes. So the first thing, we design the third tools, which called the graph optimized graphic correlation. And my student Zhenbo Lu will introduce this method in details. So in general, you can see that this is a horizontal axis, which is a composite sequence generated by a CONOP calculation, and we can take it as a standard, and we can calibrate, for example, IODP geocal with a fossil occurrence and correlate it with a CONOP generated composite sequence with this method, which of the graphic correlation. And you can see the age depth model has been illustrated as a red line over there, and also there is also some optimized and also some other solutions for the line of correlation over there, indicated by different colors like a blue color and a dark color. But most part of the section are quite consistent based on this method. And use that method, we can study, for example, for some ecological or the paleobiogeographic studies. For example, this is an example for the observation. So we can calibrate our CONOP generated high resolution time scale for the observation and early serial. And we can plot about 900 sections into this composite standard and make a detailed paleoecological studies with a temporal resolution about 30,000 a year. And so that's one solution we introduced and designed by my student Zhenbo Lu. There is also another method available. We just made it early this year. It's called the horse, because for the CONOP can only use the data. For example, this is a range chart of one section. So you can see there are many fossil records from that section. And for example, for this species, it has 1, 2, 3, 4, 5 different occurrences from that section. And the CONOP only use the first appearance and the last appearance for the calculation. So it didn't use the 3 intermediate fossil occurrence of that species. So that means after the calculation, you will be able to get the global first appearance and the last appearance of that species, but you have no idea what happened between the first appearance and last appearance, how many occurrences can be found between its range, and also what is the local observation. For example, the local observation from this section is that how to correlate it to the high resolution time scale. I mean, for the CONOP program, you cannot do that. So there is another solution that maybe we don't sequence the first appearance and the last appearance of the species. If we sequence the occurrence or the local observation, if we try to sequence them with a similar method from CONOP, then we can, after the calculation, after a CONOP calculation, we can get all the local observation calibrated into the high resolution time scale. So that's the basic idea. So it tries to sequence the collections. That's the combination of the local observation rather than first appearance and last appearance of the fossils. and then they can be used for the regional and also the global stratigraphic correlation, but use a different method which called the horizon sequencing. So here, this figure shows the performance of the CONOP and the new program. So you can see that both of them, they have the ability to quickly find a good solution. For example, after about 200 or 250 iteration of calculation, both of them, they can reach a similar good solution. But you can see that the horse, the new program, can even get a slightly better solution than the CONOP program. But both of them, they can reach a relatively good solution with a limited time. And this chart shows the performance based on the range chart of three different approach, including the CONOP and the new program horse. So you can see some of them, some of the taxa. This range chart contains about 130 species. And you can see a few of them, for example, this one, and this one. Three of them, they show significant difference among those three methods. But in most of them, their first appearance, they become quite consistent. And we do some test evaluation on them, and they are significantly correlated with each other. So the, in the past 10 years, we try to design some program which can help us to improve the resolution of our geological time scale and use it in the regional and global correlation. And we think that indicates one important pathway to the new era that's data-driven or the data-intensive scientific discovery. So this is why we try to establish our big data platform, and we try to design the new algorithm from the computer science into our geology to solve our geological question. And so that will be the future directions we will keep working on and try to make some more progress in the future. So this is a group contribution by my team. So here shows my colleagues and my students from the Nanjing University, and so that's my talk. Mike.
Great. And we're following up with two more small talks. Is that right?
Yes.
Okay. I think we'll have questions at the end. That's okay with everyone.
Hello everybody. Can you see my screen? The shared displaying PowerPoint.
Yeah, we can see
Thank
it.
you. Okay, thank you. Hello everybody. I'm Zhengbo Lu from Nanjing University Professor Fan's team, and today it's my great honor to give you my presentation about graph optimized graphic correlation, GOGC in short. And this is an automated framework for combining the stratigraphic data. To begin with, let me introduce a little bit about the background of my research, totally about the graphic correlation. Let's start with the conflicts in the traditional biostratigraphy, also displayed in Professor Fan's slides. Here, as shown in the left figure, given two sections with 5 shared Texas ranges in different colors, there is a very important problem about how to correlate those two sections based on those 10 bioevents. The simplest way we could do is just link those 10 bio events. However, we may get this tangled pattern like this. And this is generally because the events are diachronous in different locations because that means the bio-events occurred in different time in different locations. And there can be many reasons for the diachrony, like the spatial and the temporal heterogeneity, which means, for example, one species after its origination needs some time to go from one location to another. And also the preservation unevenness, sampling biases will also worsen this situation. In traditional biostratigraphy, people may prefer to remove those discordant events and regard the left concordant events as markers. However, we know that this process is subjective and sometimes doesn't work in some locations. Also, it would severely reduce the resolving power of our biostratigraphic data. Like here, we only have 5 biostratigraphic events compared to 10 initially. In 1964, Shell put forward a new method named graphic correlation to solve this problem, where he used a two-axis plot to represent all the biostratigraphic information before solve the diachrony. So here we could put the section in the graphic correlation, we could put the section 1 on the y-axis and the section 2 on the x-axis. And then since we already know the depths of each bio-events in each section, we could draw them into these two-axis plots like this. And the most important step of the graphic correlation is to find out a line of correlation to show the temporal consistency between those two sections, as shown by the black line in the right figure. After we get this line of correlation, we could then use it to adjust the incomplete ranges against the LOC so that corrects for the regional diachrony instead of removing them in the traditional biostratigraphy. For example, here we could found that this brown first appearance seems to be occurred in a too high level in the section 1. We could then move it downwards. This is equal to extend the ranges of the brown species in the section 1 like this. And also graphic correlation can generate a composite standard by first we could select the section 2 as a standard reference section and then integrate other stratigraphic information from the section 1 into it, making it a composite standard. And this composite standard would contain the most complete stratigraphic ranges than any other local sections in our dataset like this. Although we know that the graphic correlation is extremely powerful, it is still challenging to determine the line of correlation. In the early time, people may prefer to draw the line of correlation in a qualitative way, like the splitting basis and tops put forward by Millers in 1977, where there are 3 rules. First, people are allowed to draw the line of correlation with the slope in any segments to be non-negative. Instead, it would violate the law of superposition. And also, the economy of fit requires the LOC to contain as many biotic events as possible, so it can account for as much observed data as possible. And finally, if we have a composite sequence in the graphic correlation, the LOC is supposed to split the bases and the tops above and below. This is to assume the reliability of the composite standard. Later, some researchers trying to find out some quantitative way of doing the graphic correlation, and most of them apply the regression analysis, where they would use the fitting line in the regression analysis to be the LOC, so that the LOC can best fit the original data points with the least fitting residue. In other words, the deviation between the line of correlation, the fitting line, and the data point would be the least. However, this method can only generate a single straight line, which is nearly impossible in the real segmentation rate, because we know that the slope of the LOC represents the accumulation rate, which cannot be always unchanged throughout the time. To improve this, some researchers try to use a multiple line segment to better fit the line of correlation as shown in the right figure. However, there's also some challenges like how many segments we need and determine where to place those segments. Fan et al. Also designed a method to solve this problem named Sinocore, where this program allow the user to select the first or last appearance points they believe on the screen, and then the program will generate the segments and the LOC based on those segments through the user selected points. However, this method still requires manual point selection and sometimes can be a little bit time consuming if you have lots of sections or sites in your dataset. And also, the selection is a little bit subjective that may affect our results. In contrast, Zhang and Plotnik in 2006 tried to utilize the genetic algorithm, GA in short, to determine the optimal number and positions of LOC segments. The main process of their approach includes first generating an initial large set of the possible LOC segmentations, and then the program would evaluate the fitness by ranking the LOC segments using the least squares for best fit. And finally, this fitting would be done many times until the fitness no longer improves. Their methods actually have, to some degree, can draw the line of correlation in a more objective way. However, there are still some limitations. First, the GA, the genetic algorithm, is one of the heuristic approach, so it can only approximate a potential optimal fit. And also, the performance of this algorithm is highly influenced by the parameter tuning, like the mutation rate, crossover rate, and population size. So in our study, we tried to find out and designed a new way to automatically determine the LOC by using the graph optimization. To briefly introduce, the graph theory is a branch of the mathematics that studies the graph as shown in the right figure, which are structures composed of the vertices, as shown by the blue nodes here, and the edges link them. Sometimes the edge can be directed, which means you can only go from these nodes to another in this way instead of going back. And the graph based on these directed edges is called as directed graph. For a directed graph like this, there is a very interesting problem about finding out the longest path. And this can be very easily done in this very simple graph as shown by the red lines here. However, this process can be a little bit sometimes very difficult if we have a more complicated graph. Okay, so let's now go back to our stratigraphic correlation problem. So here what we wanted to do is to apply the graph optimization to solve the problem in stratigraphic correlation, or specifically the graphic correlation. So the first step we need to do is to extract an objective of graph from our stratigraphic correlation problem. So here we define the graph G. In the G, the vertices, each vertices represents a stratigraphic event, like the first appearance or the last appearance, and the edges would occur between two nodes or two stratigraphic events only if the latter occurs no lower than the former in both correlated sections. By doing so, we can then transform our stratigraphic correlation problem into a well-structured format before we reformulating our LOC estimation problem as finding out the longest path in the graph G. As I mentioned above, finding out the longest path can sometimes be very difficult because if our graph is very complicated. So, however, here, thanks to the transitive and monotonic positional relationship among our stratigraphic events, it can be very easily solved. For example, given Biotic events A, B, and C in both two sections, if event C is higher than B, B is higher than A, then we can imply that the C must be higher than A. And the graph constructed based on these relationships should be a directed acyclic graph, DAG. For a DAG, the shortest and the longest path can be efficiently found by topological sorting and dynamic programming, as shown by the serial codes on the right. So here we did a comparison between our generated LOC based on graph optimization with that generated by the Zhang and Plotnik using the genetic algorithm. In the right figure, we draw the Zhang and Plotnik's LOC in the dashed line and our LOC in a solid line. And we could found that the shape of those LOC generated by two methods is generally comparable in trend, but differs in specific details. Like here, there is a key bed of bentonite. Zhang and Plotnik's LOC doesn't necessarily pass through the original data point, so it seems to be more likely to be influenced by the outliers near to this key bed of bentonite, so the LOC doesn't pass through it. However, for our study, the LOC strictly adheres to the original data points, so it is not very sensitive to this outlier. Next, we use our generated LOC to finalize the process of graphic correlation, that is to composite the stratigraphic events into a composite standard. The workflow includes, first, we would input the original section data into the program, and then we will rank all of those sections. In other words, we are trying to determine the order of integration. And there could be many rules of ranking, like based on the events count of the section, the number of collections, etc. And finally, we will select the highest ranked section as the standard reference section before we iteratively integrate the sections, the biostratigraphic information from other sections into it, making it the final composite standard. And finally, we will generate the best composite standard with the most complete stratigraphic ranges than any other local sections in the dataset. Here we did a validation of our generated composite standard by first we test the robustness of it to the sorting strategies, where we generated 6 composite standard based on 6 different rules of sorting. And the final result shows that they are very highly correlated with each other with a correlation coefficient higher than average in 0.96. So that means overall the GOGC is robust to the sorting order, although sometimes there would be slight influence. And also we compared our generated composite standard with the composite standard generated by the CONOP, and the average coefficient is nearly 0.9. That means our GOGC-derived CS is consistent with CONOPs. However, we also find a very interesting phenomenon, that is the CS generated based on the prior knowledge sorting shows better alignment and lower deviation than the random sorting. And also here are some promising uses we see for our GeoGC program. First, also has been introduced by Professor Fan, we could use our GeoGC to apply any existing composite sequence to do the stratigraphic correlation. For example, here we use our longest path LOC to correlate a local section named Honghuayuan with the composite standard generated by the CONOP. And based on the LOC, we could identify the hiatus or the condensed interval in the Honghuayuan section, because we see there is a nearly horizontal line of correlation at around 15-meter depths in the local section. Also, we could assign the rescaled composite depths from the composite sequence back to our local sections for every stratigraphic event, so that we can apply a directed correlation with other sections across the same basin based on this unique composite standard. Also, we could use the GeoGC to refine the existing CS when sometimes we have some newly discovered sections. For example, we can also generate a line of correlation like this so that we can add the new event from the sections into our composite sequence. And this process can avoid some computationally expensive reruns. Like for the CONOP, it requires the high performance computer to do when you have so many thousands of sections in your dataset. And another use is that recently we are trying to apply our GeoGC in an International Ocean Drilling Project (IODP) to generate the age depth model construction based on as shown in the right figure, where we correlate one core from IODP site U1555 directly to a calibrated timescale, that is the GTS 2020 generally. And our longest path LOC can help to filter out those diachronous bioevents before we build an objective and reproducible age depth model. And this one is thought to be a great progress in the age model construction than previous one, because the previous one is generally constructed based on manual method or qualitative method. However, our method would be more quantitative and objective. And this program is also very promising for large stratigraphic database, like the one stratigraphy and GBDB, PBDB, because we know that the time scale or even the time points may change throughout, maybe changing throughout the time. And so if there is a program like this, we could apply it to those databases to dynamically recalibrate it, the ages for the data in the database, so that we could keep every data from different localities synchronized, and this would finally highly support the large-scale study, including the life evolution based on correlation and climate change. Okay, to conclude, in our study, we have developed a graph theory-based method, GeoGC, to automatically do the graphic correlation. And this is actually done by generating the longest path LOC based on the longest path problem in a directed graph using the topological sorting and dynamic programming. And we found that our generated LOC is comparable to previous best fit LOC, but in a more deterministic way, and also robust to outliers. Also, the composite standard derived by GeoGC is robust compared to the CONOP, but sometimes the sorting strategies may matters. And finally, we also we could see some promising broad applications of our GeoGC in fields like applying and refining the composite standard and automated age depth plotting. And this is all of my today's presentation. Thanks for listening. Questions are welcome.
Thanks very much, Chengbo. I think we'll have questions after the third talk, so that we can cover all three talks at once. Otherwise, we'll be very much over time. So can I introduce the third speaker? I think it's Tianyi Chu.
Yeah, can you see my screen?
Yeah.
Okay, thanks, Mike. Hello, everyone. I'm Tianyi Chu. I'm now a master student from Nanjing University, and I'm really happy here today to share our recent progress on the quantitative stratigraphy method development. We spent around 1 year to develop this method named horizon sequencing. We call it HORSE, which has the ability to harmonize regional and global stratigraphic records for geological timescale and evolutionary study. So generally speaking, how do we know the age of fossils? Here we have a list of fossil occurrences from the paleobiology database, or PBDB. We often take the age information for these fossils for granted, but where exactly do these ages come from? Are they from the fossils themselves? Not likely. Usually, these ages are inferred from the rocks that contains the fossils through stratigraphic correlation. This involves various methods like lithostratigraphy, geochronology, biostratigraphy, chemostratigraphy, and so on. We have different ways to do stratigraphy correlations, but fossils or biostratigraphy is preferred in Phanerozoic study, especially paleontology. For a balance of uniqueness, universality, and isochrony. In terms of biostratigraphy, we look at the similarity in fossil composition between different sections by comparing which fossils are found, where we can correlate rock layers and infer their ages. But when we do biostratigraphy correlation, we often assume taxa appear and disappear at the same time globally, but that's rarely the case. Due to biogeographic provinces, environmental differences, and the sampling bias, the same species might show up at different times in different locations. These diachrony can lead to misaligned biozones across sections. So it's crucial we account for these factors when interpreting global stratigraphic patterns. Traditional biostratigraphy relies on index fossils to define biozones. They tend to give us the least diachrony, but in reality, biozones can be limited. They are not always present. Their resolution is low, and they may not be perfectly isochronous. So the key question becomes, how can we make better use of other non-index taxa? Given the limitations of traditional biozones, it's important to take a more quantitative approach by recording the first and last appearances of taxa across multiple sections. We can build more objective correlations. While probabilistic methods estimate the average ranges, the deterministic approaches aim to reconstruct the full evolutionary signal. And if our goal is to capture real evolutionary history, deterministic methods offer a more robust framework. The graphic correlation introduced by Shaw in 1964 gave us a visual way to correlate two stratigraphic sections by plotting the first and last appearance of taxa. The goal is to find the line of correlation, or LOC, that minimizes the range extension or misfit. Between the sections, it has strong theoretical grounding, and in practice, it's often subjective and quite inefficient. Still, it helps us see where discrepancy arise and can guide more robust quantitative methods. To address the limitations of graphic correlation, CONOP was developed as a more powerful, multi-dimensional alternative. It applies constraints like coexistence and range overlap and uses optimization algorithms to minimize total misfit across multiple sections. Instead of comparing just two sections, it builds a composite section that integrates all available data. This gives us a more global, consistent, and statistically rigorous correlation. Now, the challenge is, how do we actually find the best composite sequence? Since the number of possible event orders increases exponentially, CONOP uses an approach inspired by the traveling salesman problem. It applies simulating annealing, which introduces small random changes and evaluating whether to keep them based on a cooling temperature function. This helps escape local optima and move toward a global minimum misfit. Once we have the composite sequence from CONOP, we can calibrate it using the index fossils with known absolute ages. This allows us to interpolate the ages of all events across the sequence. As shown here, this time-calibrated framework can be used to reconstruct global biodiversity patterns through the geological time. The figure on the right from Fan et al. 2020 shows how species and genus richness changed through time. These kind of analysis help us quantify evolutionary trends at a global scale. While CONOP offers a strong global framework, we increasingly need methods that capture local patterns like spatial heterogeneity, sampling biases, and within-region community shifts. As shown on the right, spatial distributions of fossil occurrences and richness can vary greatly, and approaches like minimum sampling trees, spanning trees, the MSTs, are now used to correct these biases. But CONOP only address these indirectly and often with additional bias. So HORSE builds on the horizon annealing algorithm, which was originally designed to preserve local stratigraphy resolution by sequencing horizons rather than discrete events. While CONOP focuses on the global event order, HA introduced horizon-based optimization using simulated annealing. HORSE generates and significantly improves HA by incorporating more powerful optimization methods and a formal composite of horizons. This allows it to match CONOP in event resolution while uniquely retaining local fossil occurrences. To improve both accuracy and efficiency, we redesigned the core components of HA when developing HORSE. We replaced score-based mutation with direct horizon switching and introduced secondary penalty updates that respects the negative evidence, such as taxon is implied to occur locally but is not actually observed. On the optimization side, HA moves beyond simulated annealing by using genetic algorithm and parallel computing, enabling fast and scalable performance on datasets with over 100 sections. Once HORSE produced the composite of horizons, we can also calibrate it using indexed fossils and interpolate absolute ages across the sequence. Crucially, each horizon retains its original location, lithology, and local taxonomic data, allowing us to build an ordered stratigraphy sequence that is both time-resolved and spatially explicit. This enables us to move beyond global summaries and investigate the regional patterns, such as taxon richness and biogeographic dynamics. As shown here, we can track how species distributions shifted over time during the Gobi, with full control over spatial and temporal resolution. This level of detail isn't possible with CONOP or most event-based methods, making HORSE uniquely powerful for paleogeological and macroevolutionary studies. As for the algorithm design, HORSE use a parallel genetic algorithm to efficiently explore the space of possible horizon sequence. It starts with the population initialization and applies multiple crossover strategies like PMX, OX, CX, etc., and combines local and global mutation operators. Finally, the tournament selection ensures the fittest solutions are retained across the generations, all running in parallel to boost speed and scalability. To evaluate HORSE, we tested on both simulated and real datasets. The simulated data come from a birth-death model with preservation and sampling biases, while the real data includes Cambrian marine fossils and graptolites. For most of the method comparison, we use dataset 1, the simulated dataset with short taxon ranges across 80 sections. We first compare the base performance of CONOP and HORSE. We could see that both methods show good agreement with the true taxon order, but HORSE is already slightly more accurate as seen in the tighter range distribution. Notably, CONOP tends to overestimate taxon durations with a much higher mean duration than the true value. HORSE shows milder inflation, indicating its structural advantage in controlling implied range extension, even without additional constraints. Then we add the teaser, the secondary penalty, to account for negative evidence. We could see that both methods improve substantially. Their computed taxon ranges become much closer to the true values, both in shape and mean duration. Notably, CONOP shows a larger improvement, reducing its earlier overestimation significantly, while horse still performs well. We also tested how consistent each method is under repeated runs using different initializations. As shown here, CONOP without teaser is highly unstable in the light orange dot, producing scatters results across runs. In contrast, horse with or without teaser, the cluster tightly and stays close to the true sequence. The teaser clearly improves robustness for both methods, but horse is consistently more stable and robust overall. From the perspective of parsimony, horse is fundamentally simpler than CONOP, while CONOP requires 4 separate steps: extracting, sequencing, spacing, and locating. Horse performs all of them in a single unified step. That means fewer assumptions, fewer tuning parameters, and fewer chances for user to induce bias. As a result, Horse is also more flexible for incorporating diverse stratigraphic data types. To summarize, Horse is a promising method that combines the global accuracy of CONOP with the local resolution of HA. Its key advantages is the ability to preserve spatially explicit horizon-level fossil information could allow us to harmonize global and regional records in a single framework. Technically, HORSE is designed to scale from structured databases to parallel computing on GPUs. This shift greatly boosts its efficiency and opens the door to real-time, large-scale applications. That said, the method is still under development, and broader testing across dataset is the next critical step. Besides, one of HORS's most powerful future directions is its ability to integrate different types of geological data within a unified temporal framework. While it currently uses fossil records alone, it can also incorporate like ash beds, geochemical signals like delta C13 excursions, or radiometric dates by assigning penalties directly to horizons. These flexibility enables sequence slotting, where different stratigraphy records from cores or regions can be compared numerically. As shown here, both biotic and abiotic signals can be aligned and analyzed in parallel on the same timescale. This opens the door to richer multi-proxy reconstructions of Earth history. Building on its ability to align diverse geological records, HORSE also opens up new possibilities for investigating the relationship between biodiversity and environmental change. Past studies often relied on global correlations between species diversity and proxies like CO2 or temperature. But with HORSE, we can now align fossil and environment data horizon by horizon or cell by cell, making it possible to explore spatial heterogeneity and causal mechanisms in greater detail. For example, we can combine fossil records with isotope data, paleoclimate simulations, or Earth system model outputs within the same temporal framework. This enables more refined reconstructions of biogeography and more robust tests of environmental-driven macroevolutionary dynamics. If you are interested in learning more about the HORSE method, you can scan the QR code here to access our full paper. And if you'd like to apply HORSE in your own research, or if you have any questions about the method, please feel free to contact us via email. We will be happy to share the source code and help with implementation. And that's all for my talk. Thank you.